All right, we are recording. So today we're going to talk about cell structures, membranes, cell transport. So there's lots and lots and lots of stuff with this. We will be getting into photosynthesis and into cell respiration a tiny bit, but we'll just kind of touch on it, do like a real brief overview of that. Because packet 3 is all about enzymes, photosynthesis, and cell respiration, the energetics. What we don't hit today, we will hit then. So when we talk about our basic cell structures, we're talking prokaryotes, we're talking eukaryotes. Your common features of those types of cells are going to have a cell membrane. Some of them will have a cell wall. They are going to have DNA, but the DNA is going to be organized differently. Both of them have ribosomes. However, the ribosomes are also different. And the size of them is different as well. Prokaryotes are much smaller. Prokaryotes are about the same size as mitochondria and chloroplasts, which is then going to be a link to our endosymbiotic theory, which we're going to be talking about later. And the actual cell membranes, even though they both have them, they are chemically slightly different. We're not going to talk about what those differences are. Just know that they are slightly different in their cell membrane construction. Also going to be important for our endosymbiotic theory. Things that are important differences with our DNA, with our prokaryotes. Prokaryotes have one main chromosome that is circular. That is important because then they do not need telomeres. And why do our eukaryotes that have linear chromosomes, they do need telomeres? Why is that? Maybe a better question is, what is a telomere? Right. It's all the nonsense DNA at the end of the chromosomes. And why do we need those on our chromosomes?
Because it doesn't match us perfectly.
Right. Every time it replicates, our DNA gets a little bit shorter. So if you have a circular chromosome, then that is not necessary because it does not get shorter. Prokaryotes also have those tiny little circles of DNA that are plasmids. They are 3 to 24 genes. And of course, those are the things that we utilize in DNA technology to do gene recombination. And when we look at the genetics of mitochondria and chloroplasts, they have circular chromosomes and they have plasmids, just like prokaryotes. Other things, ribosomes are structurally different. In their molecular sizes. That is helpful when you're doing DNA analysis of different kinds of cells, whether you are analyzing ribosomal RNA or the genes that make them, but it's also important for antibiotics because a lot of antibiotics target prokaryotic ribosomes, and since we do not have those, it doesn't affect us. Eukaryotes, a very important thing is that they are compartmentalized. They have these membrane-bound structures, ER, rough ER, smooth ER, Golgi apparatus. And the important thing about compartmentalization, which is question number 2, is that allows specialization and greater efficiency. So imagine that the second floor of Weeks was one big room. And at the same time, we're teaching AP Bio, we're teaching 8th grade science, we're teaching 7th grade science. We are teaching General Chem, AP Chem, and Honors Chem, all in one big room. I mean, you all can barely pay attention in one room. So that would lower our efficiency, that would lower productivity. And so what compartmentalization does is that raises these things, especially in terms of energy production. So especially when we take the mitochondria, that becomes much more efficient when it is brought in and has a second membrane around it. And then greater amounts of energy is going to then allow the eukaryotic cells to get bigger. And that's why they can have a larger surface area to volume ratio than bacteria because of this compartmentalization. Your basic cell parts and what they do: nucleus, your DNA replication, transcription, and RNA processing, rough ER for protein synthesis, for things that are going to be either secreted or part of the cell membrane. Smooth ER, the main thing, storing calcium for nerve transmission. So when the nerve impulse gets to the end of the synapse, that's going to allow the calcium to rush in. And then we also have the synthesis of lipids, hormones, and the cell membrane. Other things, detoxifying things, but those aren't the main things. Golgi apparatus, sorting and modifying, mitochondria, chloroplast, for cell respiration and photosynthesis. But the thing in both of them that is common is that they both are going to generate hydrogen gradients for making ATP, help power cell functions. And then lysosomes, we talked briefly about those. When a cell ingests something, so maybe a white blood cell eats a bacterium, then the lysosomes are going to digest that bacterium to get rid of it. And then vacuoles are going to be for storing things. We're going to see our vacuoles. These are mainly in plants. And that's going to be part of our, the way that plants support their tissues as a result of osmosis. Yes?
It has a ribosome on the top. What?
Oh, protein synthesis. All right, good, almost. All right, so then going to our endosymbiotic theory of how did cells become eukaryotic. First thing, we have our ancestral prokaryote. This starts to get bigger. And as cells get bigger, we're going to talk about surface area volume ratio later, but the cell membrane gets a lot more wiggly. So if we remember when we started our review and we made our artificial membranes, the big, giant membranes were all kind of wibbly, and you could blow a bubble into, blow an artificial membrane into that other membrane because it was so loose. So the same idea here, as that membrane gets bigger, it starts to fold in on itself. And then this is how this folding in right here, that's how that pinches in, and that becomes our nuclear membrane and our rough ER and our Golgi from the infolding of the cell membrane. So that's how those things are theoretically thought to have come about. But then more importantly is the symbiotic part, when that cell engulfs an aerobic bacterium. And what happens when this comes in, right here, this has one membrane, but when it comes in, it gets the cell membrane wraps around it, and that becomes the outer membrane of the mitochondria. And then later, the same thing happens with a photosynthetic prokaryote that then becomes the chloroplast. Yes?
So how would the ancestral prokaryote survive without a mitochondria?
So glycolysis. So because everything does glycolysis. So it would just be doing that. And then when you get the mitochondria in, that's when everything becomes more efficient. Because these aerobic bacteria right here, initially, And this basically would have been the matrix. And this would have been that cell's outer membrane. So it would have been pumping hydrogen out. And this would have been the ATP synthase. But it's just pumping it into the environment. So this is not going to be very effective because you're not able to build up a high concentration of hydrogen. But when it comes in and it gets the outer membrane of the original cell, then you get the outer membrane, and that's what allows it to build a much higher concentration of hydrogen, which then makes the mitochondria more productive, which then allows these cells to start getting bigger. And then the same thing happens with the chloroplast. And it's thought that this happened first because plant cells all have mitochondria, and then chloroplasts come in later, and these eventually develop into your plants. So our main points of evidence, mitochondria and chloroplast, same size as bacteria. The DNA structure of mitochondria and chloroplast is the same as bacteria, plasmid, circular DNA. The ribosomes have the same prokaryotic structure. They replicate independently of mitosis. So if you take a cell and you destroy the mitochondria, that cell does not make new mitochondria. It cannot replicate them unless they are already there. And also, it's very interesting, if you look at the inner membrane of the mitochondria, it has the same structure as a prokaryotic cell membrane. But the outer membrane of the mitochondria has the same structure as a eukaryotic cell membrane. So that just tells you that when it came in through phagocytosis, then it just didn't get eaten. It actually became beneficial. All right, so our structure with our chloroplast, so you don't have to draw it this complicated. You can simply draw it in outer membrane. I don't know why it's doing that. Inner membrane, and then just give a couple thylakoids. And so outer membrane, inner membrane, thylakoid stroma. And when we look at that, our main information, so your thylakoid, this is going to be for your light reaction. And then your stroma is for your dark reaction. And then we'll get into the details of those later, tomorrow. But basically, this is all thought to have been from an early type of prokaryote. And so when we look at this structure, this picture, all of this right here, this has two functions. It's going to capture light to make a hydrogen gradient to make ATP. And then it's going to excite electrons to make NADPH. And what is NADPH? You don't have to know the name. What's its job?
Electron receiver.
Right, it is going to carry those electrons, carry those hydrogens, and then both of these will be used as your energy source for the dark reaction, which is going to happen in the stroma. And that's where you do carbon fixation. That's where you take the CO2, put it into glucose. And so we will go over this again tomorrow, but photosystems gather the light, excite electrons, pump hydrogens, make ATP, gather light, excite electrons, make NADPH. All right, mitochondria, your simplified drawing. Outer membrane, inner membrane, intermembranous space, matrix. And so when we talk about the general functions of the mitochondria and what they are doing, again, we're going to have our 3 stages of cellular respiration, of glycolysis, Krebs, and the electron transport chain. Glycolysis actually is going to happen outside in the cytoplasm, and then Krebs and the electron transport chain happen inside of the mitochondria. So again, in this picture, what we see that we have the prokaryotic structure of ribosomes, we've got our DNA and our plasmids. And what is the significance of the wibbly squibblies? Increased surface area, so you can have more reaction and therefore get more energy production. So again, cytoplasm out here, this is where we're going to have glycolysis. That's going to split our sugar. Those products, which is going to be pyruvate, that is going to come down to Krebs. And then the main purpose of Krebs is to make our NADH and our FADH2, which go up to our electron transport chain. Electrons come in, pump hydrogens, make a gradient, make ATP. So the essential overall of this is glycolysis and Krebs. Harvest electrons to power the ETC, which makes the hydrogen gradient to make ATP. So when we look at like the intricate reactions of like cell respiration, they're not going to test at that detail. If they are looking at that detail, they will probably give you a diagram of it, and then you have to interpret things. So you're not going to have to know like the number of things that get produced in glycolysis and Krebs, or, you know, maybe 36 to 38 ATP overall for cell respiration. But they're really not going to, they're going to focus on more kind of larger thematic ideas. These that's what they tell us. And so 1 year they're like, oh, we decided to change it. And I'm like, okay, this is why we hate the AP College Board. All right, surface area volume ratio. Better to have big cells or small cells? Because there's more surface area to the volume. So when we look at this, a 1 by 1 by 1 would give you a 6:1 volume surface area to volume ratio. And then this, and the problem with this, just think about it this way. If we're talking about moving oxygen into the cell, the diffusion rate of oxygen is going to be the same. And so because the cell is smaller, the oxygen will be able to get to the center faster than it would in a larger cell. So having a smaller cell allows that diffusion rate, or allows that diffusion to happen into the cell at a much faster rate. There are some cells that are cubes. On the AP test, they often will give like all of these dimensions of all of these cubes. Just take the smallest one, you don't have to do any math. Unless they give you different shapes. And then you actually will have to do a calculation of length times width times height to get your surface area to volume ratio. But anything that is very long and thin and flat, it's going to have a much better surface area to ratio, surface area to volume ratio. So this goes to adaptation of animals. So we read, and if you remember the article about Bergman's rule, that down here at the equator, as we migrate to the poles, animals tend to get larger because they have a greater, or they have a smaller surface area to volume ratio. Would be like, but you just said the other one is better. Yes, for cells, that's true. But for organisms, being larger in a cold area is going to allow you to retain your internal heat better. So, and then the opposite idea over here, so down here at the equator, now here at the poles, what do we notice about the rabbit's ears?
The size, they're getting smaller.
Okay, so why would we want smaller ears on the rabbit as we go colder?
Less surface area.
So they lose less heat. And so this is actually an adaptation to get rid of heat. So I would be great in the desert, except for this part up here. So that is going to, so again, it's still about surface area. It's still about the adaptation. Here it's conserving heat by being bigger and having, and here it's larger surface area gets rid of more heat faster. So it's really the same idea, but there you go.
What would you like more? That's like
For which part?
second one when they get like shorter.
So it's smaller ears? Yeah. All right, so other ways that we see an increase in surface area. So in our mitochondria, we've got the cristae, so the folds of the inner membrane create more surface area. So you can have more electron transport chains happening, so you can produce more ATP. We also, in plants, we see an increase of surface area in roots several different ways. So when a seedling first germinates, the epidermal cells of the embryonic root grow these root hairs. So very, very thin. These are all single cells. And so they are very, very thin. So they have lots and lots of surface area to rapidly absorb water, rapidly absorb nutrients to help the seed grow. As the plant starts to get larger, then you have the roots. And then all around the roots, you have mycorrhizae, which is what? That is your relationship of fungus with the plant roots. So the fungus increases surface area to allow for greater absorption of water and nutrients. And then the plants give the fungus sugars. So it becomes a mutualism. In the human body, we increase surface area when we are breathing using the alveoli. So our trachea is going to branch into the bronchi, which branches into the bronchioles, which keep branching and branching and branching into the alveoli. Our lungs are not just big balloons, because if it's just a big balloon, the amount of surface area that we would have for a gas exchange would be very, very small. So by branching and then ending in these tiny little alveoli, which are then wrapped in capillaries, another way. So here is a picture of the majority of the capillaries that are in your hand. So you see all of that branching, creating more and more surface area for both O2 and CO2 exchange, but also the exchange of nutrients into cells from capillaries. And then in the intestines, so as your food has gone from your stomach into your small intestines have these folds. So that's one layer of increasing the surface area. On those folds, you have these extensions right here that are called villi. So this is the folds on the folds. And then on the villi, you have tiny little extensions of the cell membranes, which are called microvilli. We've got folds on folds on folds. So if you take the average adult small intestine, which is about 28 feet long, and you stretch it all out and to the complete surface area, it would be about the size of the interior of the lab, is how much space your small intestine would take up. That's to give enough surface area to absorb all the nutrients that you need. Other things for CO2 and oxygen exchange. So again, we already talked about the alveoli, all the tiny little air sacs wrapped in capillaries. And then in fish, you've got the gills, and then the gills are broken down into these little gill hairs, and each of those have tiny little extensions on them where you have the capillaries flowing through for the gas exchange. So again, folds on folds on folds, increasing more and more surface area for that gas exchange. And then just a throwback, if we remember, we've got this counter current flow. So as the water is going one direction, the blood is going the other direction, so that you get greater than a 50% diffusion rate. If they were going the same way, the maximum amount of oxygen being able to absorb would be 50% of what's in the water, because then they would reach equilibrium. But by going opposite directions, the blood is always encountering a higher level of oxygen. Therefore, you can absorb more from the water. All right, plants, our O2, CO2 exchange is going to be in our stomata at the bottom of the leaves. So this relates to surface area depending on the leaf. So if you are in the tropical rainforest, big leaves, because there's lots of water. In the desert, small, maybe no leaves. And then all your photosynthesis happens in the stem. But again, we've got our guard cells. Our guard cells are going to open when water floods in. Water floods into the guard cells when potassium is actively pumped in. That lowers your water pressure, water potential, sorry. And then that causes the water to follow that salt. That increases the pressure inside of the guard cell, and they open up. And then that allows the oxygen to exit and the CO2 to enter to allow for photosynthesis.
Can you repeat that?
Repeat all of that?
No, just like, yeah. Or, yeah.
How it opens and closes?
Yeah.
Okay, so at night, no sunlight, no photosynthesis, we're not producing energy. So when the sun comes up, then the cell now has energy to actively pump the potassium into the cell. Potassium is salt. So when the salt gets actively pumped in, water is going to follow because of osmosis. That fills up the vacuoles of the cell, and then the cells expand. And when the cells expand, they open up, forming the stomata. So the guard cells are actual physical cells. The stomata is just the space. You know, it's like, you know, when you open your mouth, you know, the hole in your mouth isn't, you know, it's just a space. And then that's going to allow for the transpiration to occur. This is where we go back to that transpiration photosynthesis compromise idea. So we have to have that open to get our CO2 in so we can make sugar. But as soon as that's open, the plant's going to start losing water. And so the plant has to balance how much sugar it needs versus how much water it can lose. So again, not a problem in the rainforest because there's always water. But when you get to drier conditions, they may have to have some different adaptations. Nitrogenous waste, things we need to know. Kidney filters it out. The structure that does that is going to be our nephron. So this whole thing is your nephron. And that is, should be after the next page. And so remember that we've got the glomerulus, which is this cluster of capillaries. So the blood pressure forces the fluid out of the blood into Bowman's capsule. And then the fluid from the blood passes through the nephron. This top part, this absorbs the good stuff. And then the blood, the capillaries go down the loop of Henle and then up on this side. Whereas the fluid in the nephron goes down the loop of Henle this way and then up that way. And then over here, what is removed from the nephron? What's removed first?
Salt.
Salt is removed first. So salt goes from the nephron into the capillary. Why is that critical? Water follows salt. So then when our blood comes over here, the water from the nephron goes into the capillary. If the water got absorbed first over here, and then over here where the salt is being extracted, what would happen to the water in the capillary? It would follow the salt out. So we want to pick up the salt first so we can pick up the water second. And that again is a countercurrent flow, which makes it more efficient. Go away, go away. All right, so, and then you've got about 1 million nephrons in each kidney to filter your blood. So you do not die. All right, okay, moving on to phospholipids. We already talked about those. We know we've got a hydrophilic head, hydrophobic tails. So when they are all put together, the heads line up together, the tails line up together, and it is this hydrophobic tail layer that is what makes the barrier that makes a cell a cell. That's what creates a separate internal environment which allow the life processes to happen, which are different from the outside. If you didn't have a cell membrane, all your cell parts would just mix with the environment, and you could not have gradients, you could not have specialized processes, and then life could not occur. And so when you put them together, you can get a liposome. This is basically just half of a cell. A micelle is when you don't have a double layer, just have a single layer. But the most important critical idea about this, which says draw a label picture. I just put that in there, cross that out. You don't have to do that. The most important thing is that this establishes barriers, which then allow for gradients to be made. What happens if you don't have gradients? Because you have reached equilibrium. Very good. All right, if you take one of these micelles, which is in water, and you throw it into oil, it will invert. Why? Because your hydrophilic things want to get away from the oil, and your hydrophobic things love the oil, so it's going to invert. All right, your cell membrane, lots of things that are happening. Of course, we've got our phospholipid bilayer. This is your barrier. Transmembranal proteins, what are these for? And that could be active or passive. Cholesterol, what does that do? Good. Stability. So glycolipids, glycoproteins, carbohydrates, all of those are what general function? All right, good. Whether it is helping hold cells together, whether it's establishing biochemical identity, this is my cell, it should stay here, or this is not my cell, I should kill it. Or part of the signal transduction pathway. And then you've got these peripheral proteins on the inside. Let's do a different shape with a circle. These are probably for signal transduction, which we'll go over in unit 4. And then you've got integral proteins. So these could do multiple functions, just cell structure, stability, or it's for transport. All right, cell transport, what are our 2 types? Passive and active. All right, when we talk about passive transport, what are our 3 types of passive transport? All right, what else? Diffusion, and then the specialized diffusion, facilitated diffusion. Active transport, we're going to have 2 types where we're simply going to either be pumping things across the membrane, or we're going to be forming vesicles. If we are pumping, there's 2 types, and this will be a little bit new terminology, primary and secondary. So primary, we're just making a gradient. And then secondary is co-transport, which we have talked about. Our vesicle transport, we've got exocytosis and endocytosis. Exocytosis is vesicles doing what? Good. And then endocytosis is bringing stuff in. Cell transport out, cell transport in. Pinocytosis is drawing in liquid. Phagocytosis is solid things. And receptor-mediated endocytosis is something very specific, which we will talk about. Pinocytosis and phagocytosis are going to be non-specific, and their difference is whether it's drawing in liquids, in solutions, or whether it's good.
There's a test on this
Tomorrow, hooray!
Isn't it an FRQ?
To be determined when I write it this afternoon.
That's just great.
You're welcome. Would it change how much information you're not going to put in your brain when you don't study later? All right, transport across the cell membrane, stuff that's easy is what? Louder, please. Small, non-polar. Examples. 2, CO2. Things that are difficult to transport. Polar. Big. Water. Charges, so ions. macromolecules, big stuff. Water is polar, but it's small. But it does move across the membrane easily. Why? Aquaporin. So they have their own specialized transport things. And so if water is moving through an aquaporin, so if it's helped by a protein, that is technically, it's facilitated diffusion, but since it's only water, it's osmosis. Good. All right, so passive transport. So we've got no energy with the concentration gradient moving towards what? When is passive transport going to stop? At equilibrium. So cells need to avoid that. We don't want equal concentrations of CO2 on the inside and the outside of our body, so that otherwise we can't get rid of it. We don't want equal concentrations of oxygen on the inside and outside of our body, otherwise we can't take it in. But you have just general diffusion. So this would be like oxygen moving from your lungs into your blood, a non-specific transporter. And this would be just an ion channel that anything could flow through. I don't know of any specific examples. Then you have things that will transport one substance in your small intestine. You've got glucose transporters to take the glucose from the small intestine into your bloodstream. And then, of course, our aquaporins for water and osmosis. Active transport, of course, we need energy. We are going against the concentration gradient. So we are building the gradient to do some type of work. So our main ones, we've got our sodium-potassium pump. Where is our sodium-potassium pump? Good. So we're going to pump our ratio of 3 sodiums out for every 2 potassiums that come in. And that's going to give us a difference in charge, which then gives us our resting potential for our neurons. Then, of course, we also have the electron transport chain. So we pump our hydrogens across, make our hydrogen gradient to power ATP synthase. So all of those require energy. They are different, however. Sodium-potassium pump is getting its energy from ATP. Our electron transport chain is getting its energy from the electrons that are found in our food. And of course, where do those electrons in our food get their energy from? Correct. So photosynthesis traps that solar energy in those electrons, which is trapped in the food. Then we break down the food, releasing those electrons, taking that energy to make ATP. Co-transport, this is that secondary transport. So what happens first is we use a active transport to make a gradient here. And then this gradient is needed, so both of these have to go through this co-transport protein together. If you don't make the gradient, then the sucrose cannot go through. Where would we see this co-transport happening? What kinds of tissues in plants? Who transports the sugars? Correct. So you'll see this in phloem cells. So that we can, and then once you get all of that sugar in here, then water flows in and that builds pressure to move the sugar solution. All right, role of cell walls is what? Structure protection. And so then what's happening here between this plant from here to here? It is turgor pressure, right? So you've got salt in here. So then water follows the salt. And then this builds up your turgor pressure, pushes on the cell wall, and then that gives the rigidity and the support for the plants. If this was an animal cell and you did not have that cell wall, what's going to happen when you have too much water entering? Right, it's going to lyse and explode. So again, same thing back to our stomata. You get this potassium going in, water follows that potassium, increase the turgor pressure, that causes the cells to swell and open, and then you can have the exchange of gases and transpiration. Cell wall composition. So bacteria, peptidoglycan. So again, that is protein and sugars. Plants is cellulose, fungi, beta glucan. That's the fancy word for chitin. Should know those three so that in case they just pop up. They probably won't ask like specific question of like, what is the cell wall of fungus made of? But they might, so then we would all know it and that would be good. Oh, good. There's just so little to get done. All right. Good. Energy, yes or no?
No.
Concentration gradient.
Against.
Against. Passive with. Examples of active transports. Sodium potassium pump. What else? Passive transport. Examples, osmosis. What else? All right. Okay, endocytosis, exocytosis. So this is endocytosis here. So you have stuff outside of the cell. So then as it's coming in, the membrane wraps around it, pinches making a vesicle. And then the opposite of that, the vesicle merges, pushing the stuff out. So that's exocytosis. So, so it's going, because it is causing a change in the structure of the membrane, that is going to require energy, which is why this is active transport. Examples of endocytosis. So when you have phagocytosis, which is eating a solid particle, examples that are important. This is when white blood cells eat bacteria or viruses. Pinocytosis, important examples. There really aren't any. Receptor-mediated endocytosis. This is when a cell has receptors. This receptor is called a ligand. And when the correct molecule binds to the ligand, that triggers it to draw it in and form a vesicle. So this is how certain things like cholesterol are removed from the body. But this is also how viruses trick our bodies into taking them into the cells. They bind to specific ligands. The cell thinks it's something that the virus is not, and it draws the virus in.
So why do you have something to do with, like, nerve signals? Is there something like that?
There is the protein gated ligand channels. So the neurotransmitter binds to that, and then it opens and lets the sodium go in. Okay, those are really tiny pictures. All right, exocytosis, important. So you have rough ER makes a vesicle, the vesicle goes to the Golgi changes the protein to make a vesicle. This fuses. So if you remember our backpacks in the parking lot, we were in the circle, then we fused, then the stuff that was inside, which was me, got released outside. And then your backpacks, which were on your back, on the outside of the vesicle, became inside the cell membrane. And so just depending on what is being made is going to determine what gets secreted. Sometimes you have constant secretion in the cell. So your body is constantly secreting mucus to trap dirt and gross stuff in your lungs. But then sometimes you get a signal, which then causes the cell to release something else. So speaking of respiratory system, if this is an allergen, pollen, it hits your cells and that stimulates them to release histamine, and then you get your allergic reaction. Other things that are important for exocytosis is going to be our neurotransmitters. So when your nervous signal gets to the end of your synapse, it causes the neurotransmitters to be released. The neurotransmitters then bind to your receptor on the next neuron that opens, and then sodium floods in, causing that to continue the nerve signal. The exocytosis part is the release of that neurotransmitter. And so it may take several nerve impulses to release enough neurotransmitters to transmit the signal to the next cell. Other examples of exocytosis that are important is the secretion of antibodies. So here we have 3 different white blood cells with 3 different antibodies. The antigen that fits binds to this one that activates this. This is going to be a B cell that gets activated into your plasma cells. And then all of these antibodies that are being released, those are released by exocytosis. And then those antibodies go into the blood, binding up the virus or whatever the antigen is. Yes.
In our nerves, why does there have to be a synapse? Why can't it just be straight up?
So we do have the straight shot ones. So those are electrical synapses. But those, once the beginning neuron fires, that signal is going to be always sent. And then with a gap, with a chemical synapse, you can have a different measure of control in how much or how little, or even blocking that synapse. And that's actually very good because that's where a lot of our pain medications work, at the chemical synapse level, blocking the transmission of the pain signal. So if everything was electrical, then a lot of our pain blockers wouldn't work. And so, all right, tonicity, that is hypotonic, hypertonic, isotonic. So hyper is what? So this is about the amount of solute, salt, sugar, whatever. So if something is hypertonic, it has more salt. So then if it's hypo, it has
More water.
less salt, more water. Iso is equal.
This is only relative, right?
What?
This is only relative.
Right, it is. I mean, you can have absolute measures of it. Like you can know what the concentration is. Right. So how is the water going to flow from hyper to hypo or hypo to hyper? Hypo to hyper, because less salt means more water. Water follows to where there's more salt. And so, of course, it's going to change. If you are an animal cell, what do you want? You want isotonic, because your membranes are not going to be able to support a hypotonic environment because it will explode. So yay, good, happy cell if you are animal, isotonic, but if you are a plant cell, you want hypotonic. And that will make the water flow in and give you that turgid support. Hyper is bad for both because it's going to draw out the water. This will shrivel the cells or crenate them. And then potentially here in hypotonic, these could lyse and burst. So what's our rule? Water follows salt. Okay, so that gets us into our water potential equation. So basically, the more water potential is going to be the movement of water. So what's going to happen if it's higher pressure? Water is going to move towards or away from the pressure? Away. So if you're squeezing a balloon, the pressure moves away from, the water moves away from the pressure. But if there's more salt, it's going to move towards it. So pressure increases water potential. Solute lowers water potential. And so then the combination of the two determines which way the water is going to flow. So when we do our math unit, we're just going to talk about this as a concept now. When we do all the math reviews, then we'll go over this equation and how to use it. So we'll talk about that later. But if you have a cell that is flaccid and you put it in water, the water has more salt or sugar inside, so that gives it a negative water potential. The water itself has a 0 water potential because water is not gonna, it's itself. And so then, because the salt makes the water potential lower, this -2, wherever they came up with that number, the water will move in towards that lower water potential. So water potential, the lower the number, the more water is going to move towards it. Yes.
Do you define water potential?
The likelihood that water is going to move from one place to another place. All right, so this all goes back to transpiration as well. The outside air is drier. So that means it has a lower water potential. So the water evaporates from the leaf, starts drying out the leaf, which lowers its water potential, which then draws water from the xylem. And then you have the cohesion and the adhesion that draws it up. So if our atmospheric water potential is -100, what happens if the humidity goes up in the air? What's going to happen to your atmospheric water pressure? It's going to go up. What will happen to your transpiration rate then? It will go down, because if it's humid out, you'll have less evaporation out of the leaf, and then you'll have a lower transpiration rate. Okay, of course, the sink, we kind of talked about this. We get the co-transport of the sugar into the phloem. What does that do to your water potential? So it decreases. So our source cell, we are pumping sugar into the phloem. So this is going to lower our water potential. So then water follows the sugar. As the water follows the sugar, we build up turgor pressure, and that pushes our sugar solution through the phloem. In the spring, then our sugar is going to go this way. Then our water will flow in, building pressure to push the sugar solution up. Let me go real. All right. The nervous system in 4 minutes. Yes.
So the water goes from
Correct. So then when you add sugar, you're lowering the water potential. So then the water follows that builds the turgor pressure. That's what moves the sugar solution through. All right, so this is our action potential. So again, we are going to have a resting potential of -70 millivolts. What establishes this -70?
Sodium, potassium.
The sodium potassium pump. So when we pump the sodium out and the potassium in at a 3:2 ratio, we are going to get this -70 millivolt level. Then you get a stimulus, and once the stimulus gets to about -55, that is the threshold. What happens if you are below that threshold? It won't initiate. So when a mosquito lands on your skin and you don't feel it, it's because it did not reach that threshold. But if it's a clumsy mosquito and you do feel it land on you, then it was able to trigger this to go. So once we hit the threshold, what happens? But the signal
Fires.
What happens for it to fire though?
Oh, like the gateway's open.
The gateway's open, so our sodium is going to come in. And then that causes what to happen? All right, and then what causes that to happen?
The charge particle change switches.
Well, then, yeah, and then our potassium flows out. So this is our sodium going in, the rising, because the internal charge is going up, because you've got lots of positive ions coming into the neuron. Then the sodium floods out, so now your positives going out, so your charge falls. And then why does it dip way down right there? What's happening? We are resetting. So we're doing our sodium potassium pump again. And that just happens in one section of the nerve. And when that happens in one section of the nerve, what does it do to the next section? Triggers the same thing to happen. So when you depolarize, so sodium in, potassium out, that triggers the next one to do sodium in, potassium out, triggers the next one, sodium in, potassium out. And then after that continues down the whole axon of the neuron, it starts repolarizing and resting. And so then that'll travel the whole way down the neuron from your dendrites up here, triggering the whole way down to your axons, and then what will happen at the axons? My 1 minute warning. What will happen at the axons?
Myelin sheath.
Myelin sheath is all along here. That's when we have what? The synapse and the neurotransmitters. So the myelin sheath, what does that do? Right, makes it faster because it's an insulator. And then at the end, we get our neurotransmitters being released, causing the gates to open. If this happens without you thinking about it, what's it called? Reflex. So, and why is it reflexive if you're not thinking about it? Where's the signal coming from? So reflexes, so when you get hit with a little patellar hammer, signal goes to your spinal cord, and then the reflex action comes back from your spinal cord, and then it continues going to your brain. So that's when you touch something hot, you reflexively move your hand, and then you start feeling the burn before, and that's so that you're not like, oh, that's really hot, I should remove my hand. So it allows you to, and so that is a protective, that reflex is protective, so you don't have to have your brain process it and slow all that down so you can protect your tissues, etc. Out of breath. All right. See you tomorrow for round 3.
Yay!
Only 6 more to go.
Oh, you're changing your attitude. You need to learn life.
By the way,